Hello everyone, my name is Jada Human, and today I'm going to be talking about the ornamental production aspect of our greenhouse management course at the University of Georgia. So for starters, I'm going to be talking about some of the plants that we grew in this course. I'll also be talking about some of the greenhouse activities that the students had to participate in. I'll also be documenting some of the progress of the plants. I did take a lot of photos during the growth period. I'll also be discussing some of the problems that I faced while growing these plants, and then lastly I'll get into a little experiment that we did with plant growth regulators and Gerber daisies. So here's a list of some of the plants that we grew in this course. I believe that this is most of them. We grew a variety of annuals and perennials. All of the plants are ornamental plants. We did have an ornamental production focus. And poinsettias and Gerber daisies were our main crops for the course, so we did focus more of our attention onto those two main crops. Some of the greenhouse activities included watering and fertilizing your plants, so each student in the course was responsible for watering and fertilizing their own plants. You did have to make your own fertilizer. We used Jack's 15515, and we also used a dosatron. We also allotted time during class to repot and pot a lot of our plants. That was more towards the beginning of the semester when we were doing a lot more potting up, and then towards the end we were focusing mainly on measuring and fertilizing. With our poinsettias and Gerber daisies, we did have to do a little extra maintenance. We took the EC and pH measurements of both the Gerber daisies and the poinsettias, and then we also sprayed plant growth regulators to both the Gerber daisies and poinsettias. As for some of the progress, I did take photos of most of the plants that we grew. So here is our succulent tray. This is about a 3-week difference between photos, and then there's about a month difference between these photos. As you can tell, these did really well, and they grew a lot compared to how they looked when they were first planted. This is another ornamental tray that we grew, also about 3 weeks apart, and then this was about a month later. Another ornamental tray. This one definitely could have been transplanted a lot earlier. As you can tell on this bottom photo, it does look a little crowded. This was a 606 tray, so the cells were a little bit smaller. So yeah, it definitely could have benefited from being transplanted earlier on. We also grew ornamental kale and cabbage, also did very well. We also grew some herbs, basil, mint, cilantro. For the poinsettias, I had 5 white poinsettias and 19 red poinsettias. This is what they looked like a few weeks after we transplanted them from rooted cuttings. And then this is a few weeks later. I believe this photo was taken after I pinched them. And then this is what they look like now. They still have not turned red because I haven't been treating them with any lights or anything, so the photo period has not triggered that response in the plant. We also grew Monstera deliciosa from seed. These germinated really well, as you can tell in this bottom photo here. They're definitely ready to be transplanted as well. And here are the Gerber daisies. We did start these off in a tray and then repotted them to 6-inch pots, but I didn't get a photo of the Gerber daisies when they were in the tray. So some of the problems that I faced were some pests. I did notice white flies on my Gerber daisies towards the beginning, but I let the greenhouse manager know and she sprayed for white flies after that, so that was not a long-lasting problem. The whole greenhouse did have fungus gnats. Most of the students had these little sticky cards with tons of fungus gnats on them. And then I had a bit of a problem with my, with just a few poinsettia leaves. They had these sort of splotchy patches on them, and I concluded that it was likely from the plant growth regulator that I sprayed on my Gerber daisies that it just got onto my poinsettia leaves, because the patches didn't spread or anything, so it wasn't an ongoing problem. Another problem would be drought damage. So since each student is responsible for watering their own plants, Sometimes I wouldn't get there in time and some of the plants would be super thirsty past the point of salvaging. So I did lose 3 plants, but out of all the plants we grew, these were the only ones that I lost due to lack of water. So as for the plant growth regulators and our Gerber daisies, we did do a little bit of an experiment. So I had 9 total Gerber daisy plants, and I sprayed plant growth regulator on 6 of them. And the PGR that I used was B9 in the spray form, so I sprayed that on all the leaves. And then I left 3 of my Gerber daisies without plant growth regulator on them to serve as a control to sort of see what effect the PGR has on the Gerber daisies and their growth. And then we recorded measurements of the Gerber daisies over a 4-week period. So as you can tell, the control group definitely did have more bolting than the treated group. Here are some photos of just the treated group, and then here are photos of the untreated group. And as you can tell, this group definitely has more flowers and taller flowers than the treated group. And then I also have some graphs to support the data that the photos also show. This is taken from all the measurements that we took over the 4-week period. So I did plant height, and then width 1, which was north to south, and then width 2, which was east to west. So I think that I highlighted most of the things that we did in the greenhouse portion of this course. But yeah, thank you so much for listening.